Hello, my name is James Boylan, and I'll be presenting the regulation of sperm maturation. Sperm, or the male gamete, is produced in the tissues of the testes. An image of the testes, a cutaway view of the testes is shown in the upper left-hand panel, while a cross-section of one of the structures found within the testes, the seminiferous tubules, is shown in the upper right-hand corner. Now, these tissues are responsible for housing and allowing sperm cells to mature, and it is through the coordinated action of a number of different tissues as well as hormonal regulation that allows for sperm to be properly produced. The testes are composed of a number of different structures. The first is a hard fibrous capsule found outside of the tissues that helps to provide protection. Within the testes are seminiferous tubules, and this is the place of sperm production. And a third tissue that is found within the testes are the interstitial tissues, made up of interstitial cells of Leydig. And these cells and tissues are responsible for the secretion of an important hormone involved in this regulation, testosterone. Sperm that is produced within the testes is going to mature in the epididymis, and this structure, which can be seen here, where I'm highlighting right now, allows the sperm to mature. And when sperm is used for fertilization and released from the body, it travels from the epididymis up through this tubing known as the vas deferens, and the vas deferens then takes the sperm through the abdomen of the male, where it will pass through a number of different accessory glands, the Cowper's gland, the seminal vesicles, and the prostate, where it will be mixed with secretions from these glands to create semen, and semen is then ejaculated out of the body via the urethra. The seminiferous tubules, as I mentioned before, is the actual site of sperm production, and it is shown in this image on the right-hand side of the screen. And we can see that the seminiferous tubule has two distinct regions. The first, highlighted here in yellow, is the actual tubule itself, and it is composed of two different cell types. The first being the Sertoli cells, which are in purple, and the second being the spermatogonia. And the spermatogonia are the cells that will actually mature into sperm cells. The second region shown in this image is found outside of the tubule, and outside this white ring right here, which is the basal lamina of the seminiferous tubule. This second region outside the basal lamina is known as the interstitial tissue, and it is made up of interstitial cells and capillaries. We're going to take a look at each of the three cell types listed over on the left, what their role is and how they function related to sperm production. Starting with the Sertoli cells. The Sertoli cells have an important role in nourishing and protecting developing spermatogonia. As shown in the image here, the Sertoli cells are these large purple regions that lay next to one another, and embedded within them are found the spermatogonia. To nourish the spermatogonia, the Sertoli cells release a number of growth factors. In addition, they also produce a couple of hormones, inhibin, activin, as well as androgen binding proteins. Additionally, the Sertoli cells also show tight junctions between adjacent Sertoli cells, and these tight junctions prevent a lot of exchange, limiting the substances that can enter or leave the Sertoli cells. And in effect, it creates three compartments within this seminiferous tubule. This first compartment up here is the lumen of the seminiferous tubule. we then have the basolateral compartment shown here in the purple region. And then on the other side of the basal lamina, the third compartment, is that interstitial compartment. The second cell type, the interstitial cells, are found in that third compartment, that interstitial compartment. And the interstitial cells have an important role of responding to the hormone, luteinizing hormone. And in response, these cells secrete testosterone into the capillaries and circulatory system. The third cell type, as mentioned before, the spermatogonia, are the cells that actually undergo meiosis. These cells are found in that basolateral compartment, highlighted here in purple. And these cells actually are responsible for becoming and producing mature sperm cells. Interestingly, these cells start near the basal lamina. And some of these cells undergo mitosis to produce more spermatogonia, while others begin the actual process of meiosis to become spermatocytes. Those that begin this meiotic division are called primary spermatocytes, and as they mature, these primary spermatocytes will migrate from the basal side to the laminal side of the Sertoli cells. And as they do so, they continue through the meiotic divisions until eventually they reach the apical side, where they remain embedded in the membrane to mature. Some of the maturation process that takes place in these immature spermatocytes, or spermatid, is the condensing of the chromatin into the head, a loss of the majority of the cytoplasm, as well as the production of a structure known as the acrosome, which is secreted by the Golgi vesicle. And this acrosome is important to fertilization because it contains a number of important enzymes involved in this process. Completely mature sperm do not actually occur or are not created here in the seminiferous tubule. Instead, once these sperm have, or these spermatid have undergone these maturation steps, they are then released into the lumen of the seminiferous tubule, and they will continue maturing as they travel through the epididymis, roughly taking about 12 days to become fully matured functional sperm cells. Regulation of sperm maturation is under hormonal control. Classically, the hypothalamus and anterior pituitary are involved in this. The hypothalamus releases a hormone known as GnRH, or gonadotropin releasing hormone, which stimulates the anterior pituitary. The anterior pituitary actually produces two hormones in response to GnRH: FSH and LH, or follicle stimulating hormone, and luteinizing hormone. Each of these hormones has a very specific target cell found within the tissues of the testes. Luteinizing hormone causes interstitial cells to secrete testosterone, which can then migrate throughout the body in the circulatory system. Follicle-stimulating hormone causes the Sertoli cells to produce a number of different products. The first is androgen-binding protein. Androgen-binding protein is released into the lumen of the seminiferous tubule, highlighted here. And androgen-binding protein binds to the testosterone released by interstitial cells to keep it in the lumen. And it is important to have testosterone close to these developing spermatid and maturing sperm cells, as well as Sertoli cells producing a number of different growth factors that stimulate the development of sperm. A third product of Sertoli cells are the compounds inhibin and activin. And these compounds help to regulate this process through a natural, or through a classic negative feedback mechanism that we have seen in the other hormonal control systems. Specifically, the product of interstitial cells, testosterone, downregulates the release of gonadotropin releasing hormone, as well as the luteinizing hormone, to make sure that levels of testosterone stay within acceptable limits. The product of Sertoli cells, inhibin, targets follicle-stimulating hormone and reduces its release, thereby regulating the activity or self-activity of the Sertoli cells so that there is not too much activity found in these cells to maintain adequate sperm production